Just a quick review of what we did in class and then our activity that we were working on. In the class before, we were talking about transcription and translation as a result of the cell signaling processes that we talked about. So signal would be received by the cell. We had reception, transduction, response, and we said some of the response could be to turn a gene on or turn a gene off. And transcription and translation are the processes of having that gene turned on and it making the protein. So we just reviewed our parts, that we had this double-stranded molecule here that represents our DNA. And then from that, we make mRNA, messenger RNA. We reviewed our letters, that if we had a C on the DNA, that mRNA would have a G. If we had a G, we'd get a C. If there's T, it would be an A. And then we had our special rule there that A would pair with U. And then we reviewed that all of this process, it takes place inside of the nucleus of the cell in a eukaryotic organism. We also reviewed that RNA polymerase was the enzyme responsible for that. Then we went and looked at, or that whole process was transcription. So then we went and looked at translation. So here's our mRNA. It's moved out of the nucleus over into the cytoplasm to this area, which is the ribosome. They have these EPA sites listed here. We do not need to know that. That's not necessary for us to know. But inside the ribosome, we've got 3 letters at a time on mRNA that are being read, and those were called our codons. And then we had 3 letters on our tRNA, and those were called our anticodons. And attached to that tRNA molecule are our amino acids. So we just see in the ribosome, that's where we're translating languages from the language of our nucleotides to the language of our amino acids to make a polypeptide chain, which can go through its folding process and become a protein. Then we reviewed the fact that we said that 1 gene equals 1 protein, even though we said that that's not always true, but we were going off of the idea that 1 gene equals 1 protein. But I wanted to show you how 1 gene could actually lead to more than 1 protein. And this goes with post-transcriptional processing. So we reviewed that transcription means we're going from DNA to mRNA. So post means after that. After we've made the mRNA, after our cells have made the mRNA, what processing happens, or how is that mRNA modified? And a couple of things happen. Our cells are trying to protect that mRNA, and it does that because the DNA is our master copy, and it has to stay inside of the nucleus to stay safe. If anything happens to it, the cell is not going to be able to function correctly. So we keep that DNA safe inside the nucleus, but the RNA can leave the nucleus and go over to the ribosome. That RNA, though, is a temporary molecule, and after a while, after it's no longer needed, that RNA is going to be broken down, and so the parts can be recycled. An example that we talked about with that, let me see if I can find the slide, in class, was talking about the production of insulin. When you eat, your blood sugar levels rise. So I've got all these black dots here in a bloodstream representing sugar. And then here is our cell with a nucleus and a DNA molecule. And we've got these receptor proteins built into the surface of the cell, and some of those receptor proteins recognize sugar. So when the blood sugar levels go up, there are more sugars floating around in the bloodstream, and then they're more likely to bond to this receptor protein. So the sugar acts as a ligand. But notice in this, the sugar doesn't move into the cell from that. That should be what happens. Sugar should move into the cell. But the sugar is just acting as a ligand. It's a signaling molecule. It's telling the cell what to do. And the response to that is to produce insulin. So through transcription and translation, we get insulin, which is a protein. And that insulin protein goes to a channel and opens up that channel, so then sugar molecules can move in. They'll move from high concentration to low concentration, from the outside to the inside of the cell, based upon the production of insulin. But when blood sugar levels stabilize, when they go back to normal, insulin is no longer needed to be produced. So the RNA in the cells, or the mRNA, needs to be destroyed. Well, how do we keep that RNA from being destroyed unnecessarily? Or how do we keep it from being destroyed before it's time for it to be destroyed? And that's this protection of mRNA that happens in post-transcriptional processing. So our first thing that we're going to look at is this poly A tail. Poly means many, and A is the adenine, or the A's, T's, C's, and G's that we've been talking about. And we're putting many of those A's in a row on the tail end of the mRNA molecule. And it can be 50 to 250 A's long. And the longer the tail, the more protein that's going to be produced, because that mRNA lasts longer before it gets destroyed by enzymes. It will last longer. And then the enzyme will come in and it's going to start cleaving these bases off one at a time. And so if we've got 250 bases to cleave before we get to the coding region of the mRNA, it's going to take longer, and that mRNA is going to be able to produce more proteins. The second thing we have is the 5' cap, and this is how our mRNA knows to enter the ribosome. So those are the protections that we have on the mRNA. But then how do we get from that one gene, one protein, to one gene, many proteins? And we can get that through these things called introns. Scientists at one time thought that introns were junk DNA, or non-coding sequences of DNA that were not necessary. But now scientists have figured out that those introns actually have a purpose. And in my picture, I colored these parts green because in the top picture they were blue, but they should have been green all the way through. Anyway, this is showing us that we have this intron part here. Circle. There's intron, there's an intron, there's an intron. And when we say non-coding, we mean those parts aren't used for the amino acids that are going to be part of the protein. And we have our step here. So this is our first mRNA sequence made straight from the DNA sequence. And then we do some post-transcriptional processing, and we get our 5' cap and our poly A tail, down here, our poly A tail added to it. But then we get even more processing where we cut out those introns. So you see in our final mRNA transcript, the one that's going to leave the nucleus, go over to the ribosome to be read, all of these orange sections are cut out. Those are our introns. They are cut out. And what we're left with are just the exons, or the coding sequences of DNA, the parts that will code for amino acids. And this part gets kind of tricky. We just want to recognize that there are introns and there are exons, and one gene, multiple proteins, depending on what introns get cut out and what exons get left in. We can actually make different protein sequences. Remember, the order of amino acids determines the protein. So if we have different coding regions, we get different proteins. All right, from there we moved on and watched this video about rock pocket mice. And we learned how to do transcription and translation, and we learned a little bit about mutations. So there's this mouse living in the desert in Arizona, and there's sandy areas, but there's also areas where there's a lot of basalt from a lava flow, and then the rocky areas were dark rock and the sandy areas were light. And so there was a mouse that had light fur, but over time scientists have seen that the population of mice have evolved to where most of them now have black fur. Well, how did that happen? That's what we were looking at. So I've got a copy of this paper in my room. You can come get it, or I put a copy on the Band app and you can see it there. So we started with this part, and we noticed that it says extracellular domain, intracellular domain, transmembrane, and then there's another intracellular domain part. And we compare that to everything that we've learned about proteins and cell membranes. So we took a look at this. There's our protein. And we said this part here is the extracellular part of the protein. So when we talk about the extracellular domains, we're talking about the parts of the protein that are going to make the outside part. And then this part down here would be the intracellular part. So that's the part of the protein that we're showing here. And then the part in the middle is the transmembrane part. Well, what do we know about that part that goes through the membrane? We know that those things need to be nonpolar because the hydrophobic tails there of our phospholipids don't like polar things. So that part in there is going to have to be nonpolar. So when we talk about extracellular, intracellular, transmembrane, that's what it's talking about. And then we learned how to do this. From DNA to mRNA is called transcription. And from mRNA to amino acid is translation. And even though we looked at a bunch of other stuff, we're going to simplify it here. So we've got TTG. If we follow our base pair rules, that would go to AAC. AGG goes with UCC. Don't forget that mRNA has U's, not T's. And we just kind of worked across. Oops. There we go. We just kind of worked across all of these and followed our base pair rules, and that was transcription. To do translation, we went to the last page of the worksheet and we found this chart, our codon wheel. We remember how to read that. We start in the middle and we work our way out. So I have to see, let me go back and see what ours were. AAC. AAC goes with asparagine. And we were just writing the first 3 letters in here. And you just work your way across. Now you have this first gene table, which is wild type MC1R gene. This is the gene that's responsible for coding for the color of the melanin in the mice that make them dark or light colored. And if you go back to the first page here, it talks about the MC1R gene, and you can read through that. It talks about them being controlled by pigments. One is eumelanin, and then we have pheomelanin. And you can see those things there. The last thing that we did while we were looking at this was we checked this DNA sequence right here. We looked at this DNA sequence from the wild type mouse, and we compared it to the DNA sequence of the mutant mouse. And when we did that, we saw that this first one says, let me switch to this. This first one says TTG, and so does this first one. They're the same. This one is the same, this one is the same. But once we get to this spot, which is amino acid 018, there's 1, 5, 1, 6, 1, 7, 1, 8. So once we get to this spot, GCG, if we look at this one, 1, 5, 1, 6, 1, 7, 1, 8. Instead of GCG, we have ACG. That is a mutation. And at the front of this page, it tells us about our types of mutations. We can have a substitution, an insertion, or a deletion. And so we're going to see what that looks like. I'll show you. Let's see if I need more room. The way I showed the class is that I wrote this word or this sentence on the board. And I chose this sentence because each of our words in the sentence is made up of 3 letters, just like our codons have 3 letters. And this is the wild or the normal or the non-mutant strain. And if I make a change to that, or a mutation, we can see that my change is right here. Instead of a C, I have an F. There's my change. This is an example of a substitution mutation. And it's a substitution, or I can tell it's a substitution because everything after the mutation stayed the same. We can read it the same. Where there was an A, there was an A. There's a T and a T, an A and an A. Right, all of my letters, the rest of the way through, stayed the same. We literally only substitute it in one spot. But what if I gave this as a mutation. Do you see how this does not match up with this anymore? So here's, oops, our mutation actually happens over here now. But none of this after the mutation matches. It's all different. And that was because I inserted this letter. There's an extra letter in there that wasn't there before. And then all of my other letters shifted over one position in my codon, or my reading frame is what it's really called. Everything shifted over one side. So we could do it that way, or we can have a mutation where we have a deletion. Like that. So now I still have cat ate the rat, but now this time everything has shifted over in this direction. And this is an example of a deletion. So on our worksheet, what we were doing was going through and finding where these mutations were. So we had one here, and we were there are 5 of them total. You got to find where all 5 are, and you're going to identify them as substitutions, insertions, or deletions. So in this one, it's a substitution because we changed it. This letter, let me see, this letter says G, and this letter says A, but all of the rest of this sequence stays exactly the same. None of the rest of that changed. The last thing we did was we looked at what effect that mutation had. Remember, proteins are made up of amino acids, and amino acids have different properties based on their R groups. They could be polar or non-polar. They could have a positive charge or a negative charge. They could be acidic or they could be basic. And those R groups determine where they can go in the cell membrane, or in the folding of the protein. And so then we know, can they be a transmembrane part of the protein because of their polar or non-polar properties. So looking at the effects of the mutation, we have to know what the amino acids are. So we're going to do this one. We had CGC, which goes to CGC, arginine. And then over here where that mutation was, instead of CGC, we have UGC. And that goes with the amino acid UGC, cysteine. So first we had arginine, then we had cysteine. Whenever we change amino acids, so here's our potential results or the effects. We have a silent mutation, a missense mutation, or a nonsense mutation. If we change an amino acid to another amino acid, it's called missense. If there is no change in the amino acid sequence, it's called a silent mutation. And if the mutation causes the protein to be cut short by a stop codon, it's called a nonsense mutation. So while you're going through your worksheet, you're supposed to be looking for 5 places where there are mutations. There was one of them. And then you're looking to see if it's substitution, insertion, or deletion. And then finally, is it silent, missense, or nonsense? We did work on this chart together. So if you have a friend and you want to see if they'll send you a picture of it. But please make sure that you know how to do the transcription and translation part. So don't just copy everything. And then we had some questions that we were answering, but then we were going to finish those questions next class period.